Baker, and this is my mom, Valerie Baker, and I'm going to quiz her on genetics. Let's see how much you remember.
Okay.
Ready?
Yeah, I'm ready.
Question 1. What was Gregor Mendel's model organism, and why did he pick that one?
Well, you told me that he actually tried a lot of different organisms, but a lot of them didn't work, so he just discarded that data. Not quite how we might think that the scientists would work, but then he found an organism where it seemed to follow a predictable pattern, what the characteristics of the offspring were. So he stuck with the P and looked at characteristics like yellow or green, or wrinkled or smooth.
Correct. And 2. What's a Punnett square, and why do you use it?
So, and we'd gone over, you taught me that with a Punnett square, like a simple example is a 2 by 2 table, and so you have the two alleles, two different parts of ways that a gene can be expressed by the one plant or person or whatever, and then the other plant or person that's being crossed with the first one, you put on the other axis, and then you can actually put whether they're heterozygotes, like they have this different expression of the two alleles, or homozygotes, and then it allows you to predict what the organism will be. So, for example, like the P, I think you told me, can you give me a hint? I think you told me that round was dominant to wrinkled.
Yeah.
Is that right? Okay.
Yeah.
So if you have a round that's big R crossed with, say, a wrinkled one that's little r, still all the offspring are going to be round, because they'll all get a big R. But if you have heterozygotes, like a big R and a little r, and then cross with a wrinkled one, little r, then you could get one that would get two little r's. And so it would have two alleles for the wrinkled, and then that would be a wrinkled P.
Yep.
Okay.
Question 3. If blue eyes are recessive to brown eyes in humans, how could two brown-eyed people have a blue-eyed kid?
Well, so if they both have brown eyes, and I don't know what the alleles are called, but say brown is like a big B, and say the blue-eyed kid is like, a blue-eyed allele is like a little b, then if both parents had big B's, or if one parent had two big B's, then I don't think they could ever have, tell me if I'm wrong, but I don't think they could ever have a blue-eyed kid.
Mm-hmm.
But and obviously neither parent is two little B's, because then they would be blue-eyed rather than brown-eyed if they had the recessive. So I think the only way that could happen would be if they're both heterozygotes. Like if even one of them is homozygote, big B, then they wouldn't have any blue-eyed kids.
Yeah.
But if one of them is a heterozygote, like Dad must be a heterozygote, big B, little b, for you to end up being blue-eyed.
Mm-hmm. Because you're blue-eyed too.
Because I'm blue-eyed, so I must be little b. Dad must be big B, little b. So even though Colin and Natalie are brown-eyed, you ended up blue-eyed because Dad must have given you the little b.
Yeah. Colin and Natalie are my sister and brother. What does it mean to be incompletely dominant or completely dominant?
So completely dominant, those were some of the Mendelian, that's how he could figure it out to see the patterns, because if you have, say, a smooth P is dominant to the wrinkled P, if you have a wrinkled allele and a smooth allele, it's completely dominant, that the P will just be smooth. It won't be like slightly wrinkled. But for other characteristics, like you told me, I think it was flowers that were either red, white, or you could go and have a pink.
Mm-hmm.
So that would mean that they're both expressed. So the dominant is expressed, but it's not completely expressed. It's incompletely dominant.
Yeah. And finally, what is epistasis?
Oh, this is kind of hard, a hard concept, but see.
Did I teach it to you well enough?
I hope so, because we did it once, like a while ago, and once more recently. So, so with epistasis, that means that there has to, the expression of the genes interact. So the expression of one gene is required for the other one to be expressed. So the example you gave me was Labrador retrievers. So labs can be black labs, it can be yellow labs, or brown labs. So if there's, I think it was black is dominant to brown.
Yeah.
So if a dog is a big B and little b, or two big Bs, you'd think they would just be black. But if there's another gene called E that has to be expressed in order for it to be any color at all. So if that other gene E is not expressed, then neither the brown or the black, forget it, neither one's going to be expressed, and the lab will be yellow. It'll be a yellow lab.
Yeah.
So if you had two little E's, then it doesn't matter whether you have a big B or a little b, or two big B's or two little B's, the dog will be just yellow, because that E gene is not being expressed, so it's interacting. And I think, let's see, epistasis means something like stop. Is it stop expression or
Stoppage, yeah.
stoppage? So if you just have the two little E's, the expression of the other ones is just completely stopped.
Mm-hmm.
Do you want to give me like a genotype, and I try to guess the phenotype of the dog?
Okay, so if the genotype was big B, little e, then what would dog's phenotype be?
Yellow, because it didn't have the expression of the E.
Yep. Okay, so Mom got all of the questions right, including the bonus question at the end.